Hello, in this screencast I'll be looking at monohybrid and dihybrid crosses. This presentation was originally put together as an eBiology lesson conducted in July of 2013. I have decided to turn it into a YouTube screencast with a private link so that it can be used in subsequent years, and also it will be accessible for anybody who was not able to download the presentation from Blackboard Collaborate. As we can see here, the picture is of a slipper orchid. I decided to use slipper orchids as the examples for this presentation because they are what I'm personally very interested in. Before I became a teacher, I grew houseplants. They were large specimen houseplants that were purely grown for their foliage. So to keep my interest, I expanded my collection of orchids and eventually ended up sort of specialising in slippers. Now, slipper orchids are a very large industry over in Europe, particularly in Germany, Holland and Denmark, where they are grown for the flowering cut flowers and for flowering houseplants. This means that the breeding of these hybrids and the selection of particular criteria and genetic traits is worth a great deal of money to a lot of people. For us, it's purely an intellectual exercise, and really we're just using them as examples to learn the principles, but there is a very real and very expensive industry behind this. The industry is always trying to go for new and interesting hybrids. The reason is that it's very much a fashion-driven industry, that new colours and textures and sizes and even flower counts on the spikes will give a particular breeder or supplier of young plants an edge over their competition. Whenever new things come along, they are going to be adopted by people like florists who will be using them for the cut flower trade. Again, because it's very fashion driven and because they can sell them more easily for a premium price. To go through the points we have down the left hand side, a monohybrid cross is where we're only dealing with one set of alleles. Now, alleles in this case we will attach to particular genetic traits. In the flowers that we can see in front of us here, which happens to be Paphiopedilum rothschildianum, and you don't have to remember that, we could be looking at things like the size of the petals or the striping or the colour of the lip, which is the red part that you can see in the front. When we're dealing with dihybrid crosses, we're looking at two different alleles at the same time, and in the particular cases that we'll be looking at later, we'll be looking primarily at background colour and striping. Later on in the presentation, I'll be looking at albinism and how orchid growers manage to produce flowers that don't have any colour to them, and we will also look at test crosses to see what a particular test subject is. Later on, I will also be looking at how various traits can be passed on from the parents to the offspring, but they're not logically being expressed to the extent that you would expect. And last but not least, I'll be looking at polyploidy, which is where we have multiple versions or multiple sets of genes. My first cross here is a cross between Paphiopedilum armeniacum, which is the beautiful yellow one at the top, with Paphiopedilum rothschildianum on the right at the top. When I'll be looking at my crosses, I'll generally be using species types. This is because we can assume that species types are going to be homozygous for all of their alleles. That means that both chromosomes have got the same allele. When we're looking at things like Armeniarchum crossed with Rothschildianum, we can see what sort of traits are going to be passed on to the offspring. If we look at the offspring, I'll see if I can get myself a different colour. Let's go for the red. When we look at the offspring down the bottom, it's a hybrid called Dihybrid, and immediately we can see that some traits from each of the parents have are dominant and some are recessive. Clearly the one that is dominant from Armeniarchum is the background colour. We can see that everything is this brilliant yellow colour. From the Rothschildianum we can see that we have huge amounts of purple striping, which we don't see in the Armeniarchum parent. There are certain traits that are really a combination of the two, but we'll come to those in the next slide. We can say in this particular cross that not only is the background colour of Armeniarchum on the left-hand side here, not only is the background colour for that one dominant, but we can also say with reasonable certainty that the background colour of Rothschildianum on the right is recessive, and that's why the offspring, which we can consider to be heterozygous, is going to show the yellow trait. If we were to draw a Punnett square for the colour, we could show the colour as C. In this case I'm going to give capital C for Armeniarchum's yellow and lowercase c for Rothschildianum's white background. So if we work out the Punnett square, we can see that we have all of the offspring are heterozygous. And because all of the offspring are yellow, we can say with reasonable certainty that Armeniarchum's colour is going to be the dominant one. And that's why I've given it the capital C's. We can say something very similar when we're dealing with the striping, where the Rothschildianum striping is dominant and any striping allele at all, if it exists in Armeniarchum, is going to be recessive. Okay, our second example, and the type at the top should say another example, not another exam. It's just the program that I'm using to put this presentation together, resizes some of the slides. We can see that Delanatii, which is the lovely pink one at the top left, gives us a lovely pink colour within the offspring, and Rothschildianum on the right continues to give us striping which we don't see in the other parent. But in this example we're also going to be looking at what traits that we would see that are neither one adult nor one parent rather than the other. An obvious example, to me at least, is the petal shape. If we look at the petals, I'll just have a blue pen. If we look at the petals, they're not as round as Delanatii's, but they're also not as long and pointy as Rothschildianum's. They're somewhere in between. In this case we would say that we have codominance. Codominance also used to be called incomplete dominance, but codominance really covers more factors. There are a couple of other traits that we can also see that are codominant. The labellum, or the lip at the front, give myself a different colour here. The labellum at the front, that doesn't show up terribly well, but never mind. The labellum at the front is sort of a combination of the two. It's much more rounded than Rothschildianum's, but is longer and slightly more angular than we would expect to see in Delanatii. If I was to draw a Punnett square for the petals, I would give ourselves our room for our 4 alleles. Now, rather than writing N, small p or big P, I need to annotate it with a superscript. In this case, the big P is for the petals and the superscript N is for narrow. Whoops, a daisy, it's moving now. And I now have the superscript, which means is the little letters at the top. I've got N for narrow and R for round. Narrow being Rothschildianum and round being Dallanatii. If we then cross these, we can see that we get PR and PN in all of our boxes. This means that the parent is heterozygous for round and narrow petals. What this means, particularly when I write it this way, is that we don't have dominance or recessiveness, but instead we have codominance. There we go. If it was big P and little p, it would show that one was dominant over the other, but it's not. So to signify this, I've had to use the superscripts, and when we see both of them together, we can see that we have codominance, and we're going to have a blend of those traits. If I saw PN and PN together, then I would know that I would have just narrow petals, but if I saw PR, then I know that I would have round petals. This comes into more importance when we start looking at my example for the albinos. So remember the superscripts and the capital letters. Just for a bit of interest, I thought I would include an interesting hybrid that was made between Armeniacum and Dallanatii. When Armeniacum was discovered about 30 years ago, growers were really pleased because this brilliant lemon yellow hadn't been available in this family at all. So it was a whole palette of colours that had been unavailable. They were excited because when they were going to cross this with Dallanatii, they were expecting to get all manner of lovely oranges and sunset colours that hadn't until then been available. The shape would have changed slightly, the lip would be slightly different, but you would still have this nice round flower. When I showed this to the students on the eBiology screencast live, then I asked them to try and predict what the outcome would be, and their predictions were very logical. It was basically the same as the commercial growers were expecting. What actually came out of this cross was completely unexpected. What the growers ended up getting was Paph armenii. It's since been called Paph armenii white, which is the white flower in the bottom. This caused a great deal of head scratching with commercial growers because they couldn't understand how you could cross a yellow and a pink flower and get a white one. And the offspring were always white. It wasn't a mistake. They were always white. Eventually they worked out that it was because Armeniacum is yellow because it suppresses every colour but yellow, and Dallanatii is pink because it suppresses every colour but pink. And when you cross the two, the offspring suppress every colour including yellow and pink, so you end up with a white flower. I threw this example in really just to show that when you're making crosses, you can't really take anything for granted. is that sometimes nature is going to throw some strange and unusual outcomes at breeders, and sometimes the mechanisms of passing things on are not always as simple as they appear. But don't worry, you don't need to learn this one for an exam. In the next couple of slides I'm going to be looking at how orchid growers manage to achieve white flowers with particularly good shape. An example of this would be looking at a freak mutation, which would be the one at the top left, which has the colour that the growers would want, and what they would want to do would be to combine it with the vastly superior shape and form of the flower that's at the top right hand corner. Now, why would growers want to even bother doing this? It's a lot of trouble to go to. Well, the reason is because white flowers are worth money, or at least flowers that don't have a dark colour to them are worth money. If you were a florist and you were creating things like wedding bouquets, you don't necessarily want to have coloured flowers. White flowers are very fashionable at certain times of the year and for certain events like weddings. So to be able to have particularly nice and unusual flower shapes and species and hybrids in the bouquet enables florists and really flower wholesalers to differentiate their product and provide something that their competitors don't. So they're worth money, essentially, sometimes a great deal of money. So in this particular case we are taking the one on the top left. We want to take that colour and combine it with the shape of the one on the right. Now the part of the flower on the right that I'm especially interested in is the large part at the back here. This large part is called the dorsal sepal. Now the dorsal is very prominent in this particular species, and it's particularly well shaped. So for a grower to be able to combine two traits that he particularly wants in a cross and derive offspring that contain both of the ideal traits that he wants is not that much more difficult than doing a monohybrid cross, but you just get a lot more outcomes and you have to throw an awful lot more away. I'll show you what I mean in this table down the bottom. Now in this one here I've the nice big dorsal, which I have outlined in the red here. The nice big dorsal here is in one of the parents, and it's this one at the top here. So I have this one I've assumed is going to be homozygous for the big dorsal. The other parent has got the small dorsal. If we just zoom back out again, we can see here that the dorsal of the one on the top left isn't nearly as well shaped as the one on the top right. So really we want the dorsal from the top right. What we also want to do is to be able to combine it with the colour, which when I've said coloured, really I'm saying that it's got the purple coloration, and the album, when referring to things like slipper orchids, album means white. So we either have the coloured or the white, and in this case it's fairly simple dominant and recessive genes. So the coloured is very much dominant to the white colours, or the colourless flowers. So I've given it a capital C, whereas the album has been given a lowercase c to show that it's recessive. So what we're doing now is we're combining two different alleles. The colour is very much dominant and recessive. The dorsal, on the other hand, is codominant. Just to make things a little bit more difficult for us, I've thrown in a codominant allele. If we look at the picture at the bottom, the large picture, we can see that the dorsal on the offspring is somewhere in between. It's not really good enough to be commercial. Really, you want the much larger dorsal if at all possible, and it is possible to do this, but it just takes a little bit more effort on the part of the breeders. What the breeders need to do to combine the albino colour with the larger dorsal is to do what we call an F2 hybrid. When we're combining two species like these two to produce offspring, we would call this the F1 hybrid. F1 really just means familial, is the first familial generation, or first filial generation rather. When we go to the next generation, what we are doing is crossing the offspring to produce the grandchildren of the original two parents. To go back to the table in the bottom corner, each of these, each of the gametes that we have here, let's just mark them again. We have the gametes at the top. These are the combinations of characteristics that we would get in the gametes from the large dorsal heavily coloured one, and the ones down the side would be the combinations that we would get in the gametes of the albino but not terribly well shaped specimen. The potential combinations that we would get in the offspring would be what we've got in the Punnett square. They're all going to be heterozygous because we're assuming that the parents are all homozygous. So the offspring are going to be heterozygous for the dorsal, and they're also going to be heterozygous for colour. What this means is that we have codominance of the dorsal, so the dorsal is not going to be small, but it's not going to be big, it's going to be medium sized. It also means that because the coloration is dominant, we're going to end up with the coloration being purple, which we don't want. However, they are essentially carriers for the albinism if we can bring it out in the next generation. Here is a great big Punnett square of what we would end up with the F2 generation. Now don't be freaked out, it's still just a Punnett square. The most difficult thing that we need to do in this case is to work out the different combinations of alleles that we would get. If we write on which allele, or what the genotype of the parent is, and the parent in this case is the offspring of the parents that we saw on the previous slide. This one is called flame, by the way, because it's neither one colour nor the other. Now the genotype that we would have would be D star, D peak, capital C, lowercase c. That's what we ended up with in the Punnett square on the previous slide. Now what we need to do is to work out what combinations of gametes we can get out of those sets of alleles. We can see that we're going to have one of each. We're going to have a D and we're going to have a C, and we can combine them in all the different ways that's possible. The first one is a star D with a capital C. We then also have the star D with the lowercase c. We have the D with the peak and the lowercase c, and then last of all the D with the peak and the uppercase c. This enables us to work out how the different alleles are going to be combining in the next generation. If we turn this into a Punnett square, we just work it through. Now if you remember, when we're a commercial grower, what we're looking for is essentially the picture on the right hand side, where we have a lovely big dorsal, which is from one of the grandparents, combined with the albino colour of the other parent. Now, if you remember, albinism does come up in the books that I've been working with, but it's generally been when looking at humans. I've been using the orchids as an example purely because I enjoy them that much more. But it still works the same. It's still a recessive gene, and it doesn't matter if we're using orchids, cats, or lions or humans. It's going to be working the same way. Now, if we work out this Punnett square, what we're going to be looking for is 2D stars. And the 2D stars means that we have the offspring in question is going to be homozygous for the large dorsal, which means that we will actually get a large dorsal. If we look on our table here, all of the D stars are in the top right hand corner, or top left hand corner, sorry. What we also want is for the other trait that we're looking for is the albino colour, which is the recessive. So we're going to be looking for 2 small c's. Now, 2 small c's are a block right in the middle of the table, like that. Where these 2 blocks interlink, and I've actually arranged them so that they do, we can see that we have the one offspring out of the potential 16 combinations. That is the one offspring that we actually want, because it has 2D, is homozygous for the large dorsal, and it's homozygous for the albino colour, which is the picture on the right hand side. We get rid of that so we can see. A flower like this is worth money. It's worth money because it's well shaped and it lacks the colour, which is why a breeder would be willing to go to the effort to produce what could end up being hundreds of offspring, only to find a handful that actually have the trait that they want. Once they have this trait, they can then interbreed those and either refine other characteristics to what they're after, or really just to produce more seeds. In one of the sacks that I've been dealing with, we've had an example of a question where 2 alleles were being combined, but one of them was for the expression of colour, and the other one was for the expression of actually having the body part in the first place. Do not be confused by this. If it's looking at 2 different things, regardless of whether or not they affect the same body part, you still need to treat them as a dihybrid cross, which is what this is, because we're combining 2 alleles at the same time. It's not much more difficult than a monohybrid cross, but you just need to remember that you have to make your different gamete combinations and then work with a Punnett square that's really 4 times the size that you would expect. A test cross is done when we're not entirely sure what a particular individual is, whether they're homozygous or heterozygous. And the way we do this is to cross them with a homozygous recessive partner. If we cross them with a homozygous recessive partner, we will get different combinations of alleles in the offspring depending on whether or not the test subject is homozygous or heterozygous. So if we think about it, what would happen if the test subject was homozygous dominant? If they were homozygous dominant, we would end up with the Punnett square at the top. where we have on the top we would have our homozygous dominant alleles, which would be capital D, capital D, and down the side we have our alleles for the homozygous recessive partner, which is the one that we would always use for a cross. All of the offspring, as we can see, are heterozygous. This means that the dominant allele will be expressed. This will always be the case. It may or may not work quite so well when we're looking at things like codominance, but when we're looking for simple dominant and recessive alleles, then this is the way it's going to work. All of the offspring will be the same and they will all show the dominant trait. However, should you ever get any individuals that show up that have the recessive trait, then we need to look at the Punnett square at the bottom. In this cross we have the same heterozygous recessive test cross subject, but we also have the one that we're going to be testing across the top. In this case we've made them a heterozygous. If we look at the offspring, we have essentially a 1-to-1 ratio of heterozygous and homozygous recessive offspring. When we're dealing with offspring that only come one at a time, then each one is going to have a 50-50 chance of being either heterozygous or homozygous recessive. If we're dealing with things like human children, it may take 3, 4, 5, maybe even a dozen children before that recessive trait shows through. But if it ever does, then you know that the heterozygous, the one of the parents is heterozygous. The more I learnt about the breeding of orchids, the more I kept coming across a particular argument that said that it made a difference which parent produced the pollen and which parent produced the pod, which one was actually fertilized. Now, to me, when we're dealing with simple monohybrid crosses, it doesn't make any sense. You have a 50-50 combination of genes, and the expression will simply be because one is recessive and one's dominant. But apparently that's not always the case. There is a lot of debate about this, and perhaps some people are seeing trends where there aren't, but there is a certain amount of science behind it. The examples that I have here are of very closely related crosses of another orchid, and the one at the top, as you can see, has a much better yellow colour than the one at the bottom. This is because the one at the top, the pod parent, as orchid growers would say, had a much better, brighter yellow colour than the pollen parent. If what we would call a reciprocal cross was made, where the pollen from the yellow, especially yellow one, was put on the ovaries or the stigma of the not particularly yellow one, then you wouldn't get such a good colour. Logically, this is due to some difference in the way that the genes are being expressed. The other trait which tends to follow the pod parent is vigour, and a larger, more vigorous plant that grows and matures faster and produces flowers that bit earlier, which again is a trait that's very important to commercial growers, that tends to be more allied towards the pod parent than the pollen parent. So there's something else involved here that is being passed on from the pod parent to the offspring that they wouldn't ordinarily get from the pollen parent, which is we would call the father really, because the ovaries are the eggs, and that's what the pod parent contributes. So what is it? When we're dealing with vigour, we need to look at what other things are inherited from the pod parent, that would potentially contribute to energy levels and vigour within the offspring. Energy levels in something like a plant would be quite reasonably affected by things like the amount of chloroplasts and mitochondria. In this case, vigour would be affected by the mitochondria, and the yellow colour that we see in the two pictures that we've got here are affected by the chloroplast, because chloroplasts produce a yellow pigment in flowers like this. In humans, we see something very similar when mitochondrial DNA, which is what is being passed on from mother to offspring, can be traced through lineages. Now, mitochondrial DNA we would write as MT DNA, and this is fairly stable across different generations. Indeed, we all contain the same mitochondrial DNA as our great-great-grandmothers and beyond. So why do mitochondria and chloroplasts have separate sets of DNA? It's because they're members of a group of organelles known as the plastids. And plastids are thought to have evolved from symbiotic eukaryotes that formed a relationship with a much larger organism, and in the case of chloroplasts they produced energy, or they produced glucose, and in the example of mitochondria they produced energy in the form of ATP. They have carried on from one generation to the next, and they get passed on when the egg gets fertilized. All of the organelles that we have in our bodies we inherit from our mothers. We inherit very little from our fathers, apart from 50% of our genetic material, and it's just the same with plants as it is with humans or elephants or fish. Because they're so stable, if they ever mutate, they can pass those traits on quite consistently to all offspring. In plants you see that with things like variegation, which is striping and different colouring on the leaves, which is caused by a mutation in the genes of the chloroplasts. And there is a whole range of fairly uncommon conditions in humans when we get mitochondrial DNA mutations. I just wanted to briefly touch on polyploidy and genetic mutations. Polyploidy is where an organism contains multiple quantities of the haploid number of chromosomes. Ordinarily you would expect an individual to be diploid, so they would have twice the haploid number of chromosomes, but sometimes they can have 3, 4 or even 5 different copies of that haploid number. When they have 3 different sets, they're called triploid. When they have 4 sets, they're called tetraploid, and then when they have 5, they're called pentaploid. There are 2 ways in which polyploidy can happen. Naturally, it can happen during meiosis, when the chromatids are being pulled apart during anaphase 2. They all end up going into one cell and none in the other. This means that you end up with essentially the diploid number of chromatids in one gamete and none in the other. If that diploid gamete is the one that is successful and ends up producing offspring, then that will produce triploid offspring, because you've got the diploid combining with another haploid. When that happens, it produces some noticeable differences in the plants. It can also be induced, which is an artificial way of producing polyploidy in plants. This is done by adding a mutagen, which is a substance that produces mutations. Generally, when we're dealing with orchids, the mutagen is a substance called colchicine. Colchicine is produced from those lovely purple flowers. It's called the autumn crocus. And what colchicine does is, if we have a look in the picture below, this is metaphase, and during metaphase it disrupts the production of the fibres, the microtubules that come from the centrioles and attach to the centromeres on the chromatids. When that happens, all of this genetic material here ends up being pulled one way and none going the other way. That gives you a cell that contains twice the amount of genetic material that you would normally expect to see. So rather than having a diploid cell, which we would write as 2N is diploid, we end up with a 4N, which is tetraploid. Excuse my writing. Now, why would a grower bother going to the effort of producing tetraploid plants? As with all of these things, it's because tetraploid plants are worth money. If we look at the green flowers, the one at the top, as we can see, we have written next to it, it's a 2N, so this is the normal diploid, but the one below it is 4N, so it's tetraploid. We can see there are some considerable differences in the size and the shape of the flower. Polyploidy tends to produce much thicker, stronger flowers, and they're worth extra money to the grower because, being that thicker and stronger, they transport better, they don't get damaged during transportation, and they also last better in the vase. So they are much more desirable. Another strong reason for using polyploidy is to enable growers to produce hybrids between things that wouldn't ordinarily cross. If the chromosome number in different species varies too much, then they're not going to produce viable offspring, or they're actually not going to set seed at all. An example would be the very interesting species we have down at the bottom here, which is a new species that was found in Peru in 2001. This one is being produced as a polyploid at the moment, so that growers are able to hybridize it with other species that it wouldn't normally be able to cross with. And the reason they're doing this is because this particular species is endangered, and they're using it to just try and keep the genes of this species in circulation so that it doesn't become extinct. That brings me to the end of this particular screencast. There was an extra slide, but I've had technical difficulties trying to get that one to work. If you do need to watch that, then I would suggest you go to the Blackboard Collaborate recording. It was mostly about, well, the one that I wasn't able to upload was on crossing over and X linkage. If you have questions about either of these topics or anything that I've written about so far, please contact me at the Edumail email account at the top, or send me questions or comments through the Edmodo page. Thank you.